We're talking about plant hormones and plant growth. So plant hormones are chemicals the plants use for communication, coordination, and development between their many cells. So functions of plant hormones include controlling plant growth and development, involved in division, elongation, and cell differentiation. The effect depends on the size of the action, the stage of the plant growth, and the hormone concentration. Hormonal signals are amplified by gene expression, enzyme activity, or membrane properties. So here's a chart of the main hormones that we're talking about. We've got gibberellin, auxin, cytokinins, ethylene, and abscisic acid. So you can see gibberellin is involved in germination, growth to maturity, flowering, fruit development. Auxin is involved in growth to maturity, flowering, fruit development. Cytokinins are involved in growth to maturity, flowering, fruit development. And abscisic acid are involved in abscission and seed dormancy. Abscission is the shedding of leaves. So auxins promote elongation and secondary growth. The apical meristem is the major site where auxin is produced. It inhibits lateral growth. It induces female floral parts and fruit. Auxins affect virtually every aspect of plant development and how they respond to environmental stimuli, and we'll talk a little bit about that in the next presentation. So the things that they influence are phototropism, geotropism, cell enlargement and growth, apical dominance, which you may have heard of for those of you taking the arboriculture class. You'll also hear about this in pruning. They're involved in root growth, fruit development, vascular development, and senescence, which is aging of plants. So they promote cell enlargement and growth. They're involved in phototropism. They're responsible for apical dominance. So the main bud inhibits the growth of buds lower down the stem. And they regulate the differentiation of vascular tissue, such as xylem and phloem. And here you see more elongated internodes here. So here's some evidence of adventitious root formation. On the left, we've got synthetic auxin. On the right, we don't. This is a African violet plant, and this is stem tissue that is rooted. So that's auxin that produces that. Okay, other ways synthetic auxins have been used. First of all, Agent Orange is 2,4,5-T. And here's a picture of them spraying Agent Orange in Vietnam. 2,4-D is one that we find a lot in weed and feed. You do see this. It is related to Agent Orange. It's not quite as toxic, but it's certainly not low toxicity. And so what it does is it prevents leaf abscission, prevents fruit drop, promotes fruiting and flowering, and controls weeds. So 2,4-D kills the plant by causing the cells in the tissues that carry water and nutrients to divide and grow without stopping. It's just uncontrolled growth, and the plant dies because of that. So I totally advise you not to use weed and feed. This is one of the main things that are used in weed and feed. It's a terrible herbicide. Okay, cytokinins move from the roots to tissues by moving up the xylem. It stimulates protein synthesis. It's made in the roots. And functions include cell division and differentiation, apical dominance, and then a delay of leaf senescence or aging. So here's apical dominance. Auxins are released from the shoot tip. They stimulate cell elongation in the stem, but suppress the lateral buds. Cytokinins are produced in the roots and can stimulate lateral buds if the shoot tip is removed. So that's why when you prune something, it may bush out, especially something that has apical dominance. Not all plants have apical dominance. But anyway, so cytokinins and auxins are antagonistic. So auxin from the terminal bud causes the shoot to lengthen. Cytokinins from the root stimulate the axillary buds. Auxin stimulates lateral root formation, and cytokinins restrain it. Ethylene, in most tissues, it's produced under stress, senescence, or ripening. The roles are fruit ripening, leaf and flower senescence, leaf and fruit abscission, or dropping. That's controlled by the separation of the plant part from the main body. And then floral sex determination in Monoecious species, it promotes female flowers. So some of the agricultural uses of ethylene are leaf abscission, hastens fruit ripening, fruit induction, conversion of flower sex expression, germination enhancement, and shorter plants. So with ethylene, the ripening of fruit is stimulated by ethylene. It's the most damaging hormone in agriculture. It accelerates ripening and causes rotting of fruits. So what it does is it involves the conversion of starch or organic acids to sugars. It softens the cell walls to form a fleshy fruit. It ruptures the cell membrane, and that causes the loss of cell fluid to form. Overripe fruit is a potent source of ethylene. You know, it's the one bad apple. That's ethylene. It promotes ripening of adjacent fruits. So in some cases you want to use this stuff. So ethylene is very important in bromeliad flower formation and pineapples, which are in that same family of plants. And ethithon is the product. And so this is great if this is all you're growing. You can certainly use this product, but you don't want to, if you're growing bromeliads and you're growing other things, you don't want to necessarily use this product. So last year what we did with our greenhouse class is we decided to try to get some of our bromeliads to flower because it had been years. They were just green. So we set up some shelves and we enclosed them in plastic, and we put slices of apples on every few plants to produce ethylene. And it took quite a while. We did this in, I guess it was September, October, and then we were able to actually sell these plants for the May flower sale with flowers. So it's a very cool way to go. It's just, it's very time consuming. That's why it's faster if you use this gas. Abscisic acid is produced in mature leaves, especially under stress. It's a precursor in roots, and then it's transported to shoots. The roles are stress response. They stimulate stomatal closure. They inhibit the premature germination of seeds. And embryogenesis, that's the formation and development of an embryo. And seed dormancy maintenance. Gibberellins, they are involved in young developing shoots and seeds. They will be involved with cell division, cell elongation. They stimulate seed germination, especially in light plants that require light. And if you don't have enough light, some of the seeds might not germinate. Lettuce in particular needs to have light to germinate. So in some cases you may use gibberellins to try to stimulate that germination in the absence of light. It also can be involved in the breaking dormancy of buds. It stimulates flowering and stimulates fruit development. So here it is in the seed. It's found inside the seed, and it actually, as I said, if a seed is having difficulty not germinating, you can see there's clouds in the sky here, not enough light, gibberellins can help with germination. So one of the things you may see are the use of gibberellins in fruit production. So on the left we've got grapes that don't have gibberellins, and on the right we have grapes that have it. And so two things are happening here. It's helped with the size of the fruit, but it's also elongated the stems that the fruit are attached to, so there's more space for fruit. So, review. Auxin is involved with cell elongation. Cytokinin is involved in cell division and inhibits senescence. Ethylene promotes senescence and ripening. Abscisic acid is involved with dormancy of buds, abscission of leaves and fruits. And gibberellic acid is also involved in division and elongation.